Again, my name is Claire Johnson. I'll be talking to you today about my experimental research here at Wrigley, investigating microbial mat and animal interaction with my professors David Roger and Frank Rossetti. First, a little bit about me. I'm a graduate student, PhD student at USC. I am in the Earth Sciences department. So what Earth Sciences is it encompasses a lot of different fields. So anything that you see in this picture pretty much. So the atmosphere, atmospheric sciences, climate change, the ocean, so anything living in the oceans is part of Earth Sciences, things living on the land, but of course the rocks, right? So rocks, geology. I'm trained as a geologist. Geology is the study of rocks and minerals and how we can use those minerals in our everyday lives. And within that geology subset are a bunch of things. So mountain building, earthquakes, volcanoes, lava, really exciting stuff like that. And also paleontology. So I study fossils. So I'm an invertebrate paleontologist, which means I study all of these creepy crawly things in the seafloor. That also includes, within paleontology, vertebrate paleontology, so dinosaurs, Jurassic Park, right? So really cool stuff. And so fossils, invertebrate paleontologists, we study all types of animal groups. So here are the 9 main types of animal groups. We study sponges is one group, corals and jellyfish is another. We have cone jellies and other special types of jellyfish. 3 different types of worms, grasshoppers. So this is arthropods, anything with a segmented body, crabs, shrimp, tasty things. Mollusks, so snails, octopuses, squids, everything like that. And echinoderms, so sea stars, sea urchins, sea cucumbers. Those, they sort of look dead, but they're really alive, right? And so that is all invertebrates, right? All the invertebrate groups. And the poor vertebrate paleontologists only have one animal group that they study, which is all the vertebrates, so the chordates. So that includes us, includes dogs, all your favorite fuzzy little creatures that scurry about on the land surface, and dinosaurs. So paleontology is this big study of past life that is preserved from us. So now we sort of know what an invertebrate paleontologist is. This guy. Is a paleontologist? Yes. Indiana Jones? Yeah? No?
Yeah.
Not? Right, he isn't. So he's an archaeologist. I get this a lot. That archaeology is the study of human artifacts and human remains. So we'll see in a little bit how the timescales of those vary a lot. So correct, he is not a paleontologist. So what does a paleontologist really look like? Yes, this is a picture of me. At a young age, I think I was doing pretty well. Starting out, I'm prepared. I have a sun hat on, got my sturdy hiking boots, collecting my trash, taking out my trash, and I seem to have a great attitude. So, as a kid, I really had led a charmed life where I got to travel all around the western United States. My parents took us around in our little Westphalia van again, and we got to visit all these amazing geologic formations that then I really had I didn't know about in the geologic way. But here I am at Mount Rainier, which is the largest volcano in a string of active volcanoes here in North America, the Cascade volcanoes. The van right here is actually underneath this grass, sitting on many, many miles thick of volcanic lava that was spewed out millions of years ago. Here I am backpacking in Yosemite, which itself was an underground magma chamber that was cooled over millions of years and is now giant mountains. And here's Cannon Beach. So this whole rock is a eroded piece of a former lava flow. So I really had no idea when I was visiting these places what the geologic significance really was, until I had to move across the country to leafy green Bryn Mawr, Pennsylvania, which is not really known for its beautiful rocky outcrops, right? There are nice rolling hills. There's a lot of green foliage, plants that are covering exposed rocks, right, which is a problem for geologists. But it's really here that I discovered geology. I was fortunate enough to be able to take a geologic tour of the campus. And so we took this tour, and it wasn't of rock outcrops, but it was actually of the building materials. So each of these buildings is composed of these metamorphic rocks with a hidden schist that was quarried nearby the campus. And those rocks were actually the center, the inside of these massive mountains that once were taller than the Himalayas in Pennsylvania, the Appalachians. So this really blew my mind. I had no idea that the connection between what the earth that we live on and our society is really so intertwined like this, so like intimately. And that was really my first taste of geology. And I had to take a geology class, obviously. It was so fascinating. And in my first geology class, I was presented with this figure. So maybe you can't read this. That's okay. It's quite dense because this is encompassing all of our what we know about the Earth in geologic time. So this is 4.6 billion years represented here. But it's not all to scale. So we have our Cenozoic, we have the Mesozoic, and the Paleozoic, and these three columns encompass all of what we know about visible life, pretty much. And then the Precambrian, this last column over here, is the rest of it. Now we'll have a little demonstration to show you really the scales of these things. Here we have one sheet of toilet paper. This is about 10 million years. So that's long. So 1/10 of this sheet is a million years. So even if I drew human history on this, the line would be too thick for us to categorize it accurately. So one sheet is 10 million years. So we have today, at the start, we unroll this. These first 7 sheets, that is 65 million years of the Cenozoic. Okay, so our first 7 sheets, 65 million years. We keep unrolling it. Is that going to stay?
I was going to say, but I'm more worried about the tissue.
Okay, here we have the start of the Mesozoic, which is 252 million years ago. Okay, we're still going pretty good. Keep rolling back. Here we go. Okay, and then this is the start of all visible life. The rest of this roll is the entire right column, is this entire roll. So if we roll this out, this is about 450 sheets of toilet paper, right? It's pretty long. So that is really crazy how much we haven't even unrolled, but we haven't even gotten to the beginning of Earth's history, right? And our time on this Earth as life, as visible life, is actually very short in comparison, right? Crazy to think about because, we live in not even one of these sheets, right? I do this with elementary school kids, and they love this. Thank you.
Oh, there goes your demo.
There goes. Delicate. So that gives you an idea of the scale, the real scale of the geologic time scale. Just crazy, right? And we're only talking about today. Okay, so when I learned about that really blew my mind. And so I obviously had to become a geology major. And as a geology major, I got to travel around the world to these exotic places. I got to go to the University of St. Andrews for a semester, which is really, Scotland is really one of the birthplaces of the discipline itself. So it's really fun to both be learning about the history of the rocks and how we date them, but also about the history of the field itself, which was really interesting to see how scientists, over the years, we've been using the same methods, but we're slowly getting to new conclusions, right? So I got to go to Scotland. I got to go to the Bahamas. The Bahamas was great. We got to learn about reefs, basically. So the Bahamas are a nice carbonate platform, very warm. Corals flourish here. So here I am snorkeling over part of a coral reef. And then here we are standing on what was once a coral reef that is now fossilized and above water, rather. So these juxtapositions were really informative for me when I was formulating my thesis work as an undergraduate in Utah, also an exotic locale. So my research in Utah, here I am, was all about how sponges, like SpongeBob, and microbes build reefs together. So they are in a cooperation in this situation where they're building relief above the seafloor. And this is an interesting relationship to study because it's actually very common in Earth's history and today. So this, again, back to our geologic timescale. Here is a more condensed version of it. We're starting at the Cambrian. We're ending at year 0, which is today. This is a chart of the trends of the history of life. So on the y-axis, we have the number of families, basically the diversity of life present at the time that we know from the rock record. So in the pre-Cambrian, it's pretty low. And then we have this big explosion in life, the Cambrian explosion, where most of our current groups of animals have originated from. And then through time, we've had these sort of ups and downs in the road, and the downs are called mass extinctions. So these are times in the history of life when many, many animals go extinct at once. And these are crisis points, right? So what happens during these crises, why they are caused, is really important for us to study in the fossil record so we can know more about it in the future. But interestingly, during these times of biotic crises in the animal world, the microbe world really has a great time. So the microbes are in reefs, are demonstrated by these scale bars here. And during low times in the animal life, you see a resurgence in the reef building capabilities of microbes through geologic time. So now we have a lot of animal life, very few microbes building reefs. And why this is not necessarily known right now. We're working on it. That's sort of the thrust of my research. Are the microbes and animals, are they fighting for dominance of the seafloor, right? Are they trying to win it out, or are they cooperating and sharing resources in the reef structure? Can they live in peace together? So it's sort of this duality between an antagonistic relationship and a cooperative relationship. And the mechanisms behind that aren't so clear. So what I'm doing at Wrigley is harvesting microbial mats, the fun spatula, at Cat Harbor, and performing experiments on them. So basically, I'm watching how the microbe mats grow and their responses to animal grazing on them. So animals come, they chew them, they move on, and what the mechanisms are behind that. So one way Oh, when I collect the microbe mats, they sort of look like this brown color. There's not much going on. But then when we let them rest in the lab for a little bit, they start blooming this iridescent green. That's how you know they're alive. So they are growing in the substrate. We add the metazoans, the animals, into the substrate. They are burrowing over time, and they may affect the topography of the microbial mat. So one way we try to measure this is using 3D modeling. So I know you guys all brought your 3D glasses with you, so you can put them on now. We won't be needing those. But the 3D modeling really helps us to take specific and very precise measurements of how the surfaces of microbe mats are changing over time in response to animal stress. So what I do is I take a ton of photos. This is a small subset for one sample, using basically a consumer digital camera. The only thing special about it is that it can take photos underwater. So you could do this in your own backyard, so to speak. Take all these photos from all these different angles. So this is like taken from the top, the sides, all of them. You try to get the scale bar in there so we can precisely measure what these mats are doing, and then I run it through my software, and it comes out with this very precise 3D model in the computer. Like there's a top view and a side view. So this is an example mound of mud that I've rendered in 3 dimensions. And this whole process, the computing process, only takes about half an hour. So it's a pretty quick, cheap, and easy way to measure responses of microbial mats, like in real time, which is pretty cool. So this is really important for having these types of methods are really important to understanding how these grow. So in conclusion, knowing what we can about the past Earth and all of the bumps and trials and tribulations that past animals have had to go through is really important for understanding what could maybe happen in the future, or even what is happening now. Because in the past, we have the full view from beginning to end of these biotic crises, right? We have when it started to go bad, we have when it was really bad, and then we have a recovery too. So there is presenting some hope that even after we are all long gone, life will continue to flourish on the planet, which is great. So with that, I'd like to thank all the Wrigley staff, the other fellows here, Vicki, Patricia, and Alexina for all their help refining my methods. And with that, I'll take any questions. Thank you.
So how do you do it? Will you take like, would you create like your own map, like Matt, like get a big sandbox, and make it start from the beginning, and then you'll take pictures of it. So you'll have your grid laid out, and you're just constantly taking pictures and see what.
Yep. And a time scale, also different. How many animals are on the mat disturbing
And are you just starting that project now? Is that what's so?
Yeah, so my mats are growing in the lab.
Right, so you got a sandbox up there, and you're.
Yeah.
How often are you photographing? Like every day?
Once they really start growing, hopefully it'll be more like twice a day. Yeah.
The microbial, diminishing microbial life, is a consensus maybe pollution in the oceans?
So
Do they have a theory?
Right. So this is a really long-term trend over millions of years.
Oh.
So yeah, not just today. But so microbes can flourish in a lot of bad environments that we think for animals. So maybe if there's more pollution, actually, they might thrive. There's a lot of different ways they can live. So yeah.
How long do your projects last? Like, how long does it go for?
About 5 days.
5 days.
Yeah, for one.
For one.
Is there anything unique about what you're finding at Cat Harbor, or is that kind of what bass is everywhere?
Yeah, so these are quickly growing cyanobacteria, which is hopefully highly analogous to what we see in the fossil record. So that's why I've chosen Cat Harbor specifically. Yeah.
Are you only using crabs as the animals, or are you using different types of
I'm
animals?
trying to get some snails to eat them too. So we'll see if that works.
How big's your mound?
Oh, 10 centimeters. 10 centimeters by 10 centimeters.
Does the software you're using have any statistical analyses abilities, or is it purely a visualization?
So the visual I showed you was actually a point cloud, and so those point clouds can be exported into things like MATLAB and R, and then you can run statistical analysis on these. Yeah, and it does have a built-in error propagation tool. Question.
So in addition to 3D modeling, are you doing chemical analysis for constituent elements?
Right, so there is, I will be microprobing some of the maps, so seeing how the chemistry differs from the top of the map to the lower parts of the map with the animal turning up. So yeah, I will be using that as well.
Thank you.
Thank you so much.